So in part 2 of this series on protein phosphorylation, we're going to talk about the effects of phosphorylation. When a protein becomes phosphorylated, what could happen to that protein? And it's going to depend on each individual protein. I'm just going to give you in generalities what protein phosphorylation could do. So there are 4 things that phosphorylation could do to a protein. The first is affect its protein-protein interaction. So in the cell, proteins interact with each other, and phosphorylation can regulate the protein-protein interaction. So here I've drawn this green protein. On the left, it's not phosphorylated, it's unphosphorylated. On the right, it has become phosphorylated. I'm not drawing the kinase in, too crowded. So what could phosphorylation do? It could affect protein interaction. So let's say there is this blue protein, protein X. Now protein X and the green protein, they have some affinity for each other, but it's a very low affinity. They hit each other, they run into each other, and they bounce off each other. They run into each other, they bounce off each other. So they have some low affinity for one another. But now that the green protein is phosphorylated, protein X has high affinity for the green protein. So now we have established protein-protein interaction due to the phosphorylation of this protein. Why did this occur? Could be a number of different reasons. Could be the phosphate group changed the three-dimensional structure of the green protein, causing it to now have amino acids exposed that interact with the amino acids in protein X. It could be that phosphate group, which is negatively charged, interacts with some positive charges on protein X, some positively charged amino acid side chains. It could be a number of different reasons. You don't have to know what those reasons are, but just appreciate the fact that when a protein becomes phosphorylated, that could lead to protein-protein interactions occurring, high affinity interactions between these two proteins. And in this course, we will see examples of this. And by the end of this course, you should know examples of 2 proteins that interact when one of them becomes phosphorylated. Conversely, phosphorylating a protein could abolish protein-protein interactions. So let's say protein X and the green protein, they have high affinity for one another, right? And you can see them there. They're three-dimensional shape, their amino acid side chains interact, high affinity. Now, when the green protein becomes phosphorylated, well, now protein X won't bind it. Maybe that negative charge interferes with the amino acids that interact with between protein X and the green protein. Maybe the 3D structure of the green protein changes. So phosphorylating protein could lead to abolishing protein-protein interactions. And again, by the end of the course, you will know examples of phosphorylation events that inhibit 2 proteins from interacting with one another. So phosphorylation regulates protein-protein interactions. That's one possibility. What about localization? That's the second thing that phosphorylation could affect. So I've drawn a cell here. There's a nucleus and the nuclear membrane. There's cytoplasm and the plasma membrane. So when proteins can become phosphorylated, that could change their localization within the cell. So here's a green protein, and it's in the cytoplasm right now. Now, let's say a kinase comes along, and the kinase becomes active, and it phosphorylates this protein. Phosphorylating a protein could change its shape and trigger localization change. So this green protein, when it becomes phosphorylated, it moves from the cytoplasm into the nucleus. So phosphorylation could change that protein localization. How did it get there? Well, we'd have to go into nuclear import signals and nuclear transport proteins, but we're not going to get into that. We're just going to say that phosphorylation can trigger protein movement from the cytoplasm to the nucleus. It could also trigger the reverse. Here's a protein, a blue protein, it's in the nucleus, and let's say there's a kinase in the nucleus that phosphorylates that protein. When it gets phosphorylated, it moves from the nucleus into the cytoplasm. So proteins can move from the nucleus back into the cytoplasm, and phosphorylation can trigger that movement. So localization from cytoplasm to nucleus, localization from nucleus to cytoplasm, phosphorylation can regulate these events. And again, in this course, we'll see examples of both of those. The last one I want to talk about, which we'll see examples of, is localization to the plasma membrane. So here is a brown protein, and that's a membrane protein. It's embedded in the plasma membrane. And this protein can become phosphorylated. But it's still in the membrane. Nothing's changed about it. But let's say this yellow protein. It has high affinity for the brown protein when the brown protein is phosphorylated. So if the brown protein is not phosphorylated, this yellow protein just hanging out in the cytoplasm, it might bounce off of the brown protein, might have low affinity for the brown protein, but when the brown protein becomes phosphorylated, the yellow protein has high affinity for the phosphorylated brown protein. So this yellow protein, normally in the cytoplasm, is now brought up to the plasma membrane when this brown protein is phosphorylated. So that's 3 different examples of how phosphorylation can trigger changes in a protein's localization in the cell. And again, by the end of this course, you will know examples of all 3 of these. Activity. Protein phosphorylation can commonly regulate the activity of a protein. So I've put the kinase back in here. This black kinase acts on the substrate here, the green substrate. So the kinase will phosphorylate this protein, this green protein. Now, this protein could be an enzyme, right? Enzymes are proteins. So maybe phosphorylating the enzyme changes its 3-dimensional structure so that it affects this protein's enzymatic activity. And we will see many examples of this in this course. So phosphorylating an enzyme can change its activity. Now, how could it change it? Well, 2 different ways. We can talk about a protein being inactive, an enzyme being inactive when it's not phosphorylated. So maybe the protein on the left, the unphosphorylated green version, its 3-dimensional structure is such that it can't bind ATP. Well, let's say this is a kinase. It can't bind ATP, it can't bind a substrate. It won't function. But when it becomes phosphorylated, phosphorylating a protein can change its 3-dimensional structure. And if this green protein is an enzyme, such as a kinase, it changes its 3D structure. Maybe now it can accept ATP and substrate, it can bind in its substrate binding domain, and it can function. So we will see in this course actually instances of kinases that phosphorylate kinases that regulate their activity. So, and we'll also see examples of other enzymes as well that can be regulated by kinases and phosphorylation. So I know that's a little weird, a kinase phosphorylating a kinase, but it happens very commonly in the cell. So phosphorylating an enzyme can activate it, turn it on, or phosphorylating an enzyme can inhibit it, inhibit its enzyme activity. So another example, which we'll see later in this course, is an enzyme where it's not phosphorylated, the enzyme is active. It can do its job, it can catalyze this reaction, but when the enzyme becomes phosphorylated, putting that phosphate group on interferes with the catalytic domain of the protein, and it can't function in its enzyme activity. So by the end of this course, you will know examples of enzymes that become phosphorylated and then activates them, or examples of enzymes that become phosphorylated and inactivate them. So phosphorylation can change the activity of an enzyme. Last topic we're going to talk about in terms of what phosphorylation can do to a protein is affect its stability. So later in the course, we'll talk about the fact that proteins can be made to disappear in the cell. Right? We got a protein around, we don't want it anymore. We don't want it to function anymore. One way to do it is to trigger its destruction. So proteins can be either stable, they'll be hanging around, they can do their job, or proteins become what we call unstable, and they become destroyed by the protein's garbage disposal, which is called the proteasome. So we will learn about proteasome function and how proteins are regulated in terms of their stability. So putting a phosphate group on a protein could cause that protein to become unstable. It can cause it to be destroyed by the cell. Conversely, phosphorylating a protein could actually make it more stable. So we'll see examples of both of these, where proteins that are made, they're not phosphorylated, they're destroyed. Whereas phosphorylating the proteins makes them become stable. So these are 4 examples of phosphorylating proteins and triggering some changes of either changing their protein-protein interactions, protein stability, protein localization, or enzyme activity. We'll see examples throughout the course of all of these. The last thing I want to mention is that proteins typically have multiple amino acids that have these hydroxyl groups. And again, in the last video, you should have looked up the 3 amino acids that have hydroxyl groups. So a protein commonly has many hydroxyl groups. Will every hydroxyl group get phosphorylated? No. So kinases only work on specific hydroxyl groups within the protein. So this black kinase up here, it's not going to phosphorylate every hydroxyl group on the protein. It will only phosphorylate specific hydroxyl groups in the protein. And usually it's ones that are interacting with its substrate binding domain and are located near its ATP binding pocket. So kinases only phosphorylate specific hydroxyls, not all hydroxyls. Can multiple kinases act on a protein? Sure, right? So this protein, this green protein, it might get phosphorylated by this black kinase. It might also get phosphorylated by this blue kinase. So this blue kinase can phosphorylate another hydroxyl on this protein. There might be a third kinase that can phosphorylate a different hydroxyl on this protein. So many kinases can act on one substrate. Will all hydroxyls be phosphorylated? No. You don't have to phosphorylate every hydroxyl on a protein. So that hydroxyl on the left there, it doesn't get acted upon by any kinases. So proteins can have multiple kinases acting upon them. The other thing that I want to talk about, because we'll see again in this course, is that one kinase can have multiple substrates. So here's the kinase back from the first video, and there is the substrate binding pocket right here where the active site is. And so in the previous example, I just showed the green substrate binding into the binding pocket in the active site. But actually, there are kinases that can phosphorylate many different substrates. And it's because those substrates all contain some motif in their amino acid sequence here that interacts with the substrate binding pocket. So there are kinases actually commonly have multiple substrates. We'll learn about a kinase that can phosphorylate 50 different substrates or 100 different substrates. So all of these proteins, all of these substrates, can become phosphorylated by this one kinase. So, in sum, these 2 videos talked about phosphorylation and how it occurs and what phosphorylation can do in a cell to proteins. We will see instances throughout the course of all of these specific examples of specific proteins and specific kinases.